Hi, everyone. Welcome to today's live broadcast, The Benefits of Bergamot-Derived Supplements in Cardiovascular Health. I'm Amber Lowry, Senior Editor of Special Projects for Nutritional Outlook, and I'll be your moderator for today's event, which is part of our condition-specific symposium. We are pleased to bring you this symposium presented by Nutritional Outlook and sponsored by HP Ingredients Corporation. I'd like to share a statement from our sponsor. HP Ingredients Corporation, founded in 2001 by Annie Eng, is a manufacturer and supplier of safe, branded, patented, science-based, clinically proven nutraceutical herbal extracts. HP Ingredients' mission is to maintain its reputation as a foremost pioneer in the global natural health industry by delivering unique ingredients backed by rigorous scientific research. HP Ingredients is a fast-growing, innovative nutraceutical company that brings to market standardized, patented botanical extracts and privately funds clinical studies in collaboration with globally prestigious universities and research institutions located in Malaysia, Chile, Italy, and the United States in support of these products. HP Ingredients' specialized services include custom product development, contract packaging services, and private labeling of cutting-edge dietary supplements and nutritional products. We have a few important announcements before we begin. The symposium is designed to be interactive, and we encourage you to ask questions during the event. You can submit questions by typing them in the Q&A box, which can be found on the right-hand side of your screen. Also, to the right of your screen, you'll find resources from our sponsor and the registration link for all 6 sessions. You can enlarge the slide window by clicking on the small square icon in the upper right-hand corner of the slide window, or by hovering your mouse over the lower right-hand corner and dragging the window to the desired size. The slides will advance automatically during the event. If you have any technical problems viewing or hearing this presentation, please click on the question mark help widget in the dock at the bottom of your presentation window. I'd now like to introduce today's speaker. We're pleased to be joined today by Professor Vincenzo Malachi. Professor Vincenzo Malachi, MD, specializes in cardiovascular and cardiometabolic research. He serves as director at the Research Center for Food Safety and Health, UMG, focusing on nutraceuticals as the field of research. Professor has authored more than 200 full-text papers about nutraceuticals and health published in top-ranking journals. Thank you so much for joining us today, Professor. Please go ahead and get us started.
Thank you very much for the opportunity to be here today, discussing on the benefits produced by bergamot derivatives in the supplementation for preventing and treating cardiovascular disease. As you know, evidence has been accumulated in the last few years indicating that cardiovascular disease produce a relevant impact in terms of socio-economic implications in approaching the problem of the global health and socioeconomic implications for healthcare. And it has been demonstrated that 1 of 3 deaths in USA are related to cardiovascular disease, and there is an increasing impact in cardiovascular deaths which are occurring under 16 years. So this means that there is an increased aggressivity of the cardiovascular impact in human health. And this is related to the progression in the major marker of the cardiovascular disease, which is represented by atherothrombosis, which you can see is a progressive disease which leads to the series of final and most impacting events which are characterizing cardiovascular dysfunction, and these are represented by unstable angina, myocardial infarction, stroke, and acute limb ischemia, and finally sudden death. So all these events are the consequence of the progression of the atherothrombosis process, which is affected by the most relevant risk factors. The final target of the activity of different stressors which are originating from both lifestyle changes and genetic conditions is represented by endothelial dysfunction. As you know, endothelial cells are the inner layer which currently releases nitric oxide, which maintains the vessel under a normal state of reactivity, and the impairment of endothelial cells and impairment of the nitric oxide release leads to oxidative stress and inflammation, which at the end generates the progression of the atherothrombosis process and produces the stroke, myocardial infarction, and the heart failure. And all those aspects are highly affected by the lifestyle, and the International Diabetes Federation indicated the metabolic syndrome as the major component which represents the target for the cardiometabolic risk. This is characterized by abdominal obesity, which is associated to elevated blood pressure, increased cholesterol, elevated triglyceride, and finally impaired fasting glucose. So all those conditions have to be considered as a consequence of the clustering of all these factors. So three of them are able to generate an increase of 2.5% risk factor for the occurrence of myocardial infarction and cardiovascular death. And this also suggested a different approach in evaluating and managing and in treating the cardiometabolic risk. As you can see here, at the beginning we had multiple independent management of risk factors. Each one was treated separately, but today we understand that there is the need of an integrated approach in order to produce integrated guidelines for the management of the global cardiovascular risk. And you can see here that the joint committees of both European Society of Cardiology and American Heart Association define which are the targets for getting protected the subjects at very high risk, which are the one which may receive a benefit in a global and simultaneous treatment of all the components. And you can see here that if you consider the LDL cholesterol as the major target for anti-lipemic strategies, you can see that the target is very precise, and you need more than 50% of the reduction of basal levels of LDL cholesterol, getting and approaching the level of 70 milligrams per ml. And if you look at the potential strategy we have to counteract the hyperlipemia and metabolic syndrome, you can see that the conventional drugs used, which are by statins, produce only at very high concentration, at more than 40, 80 milligrams, a relevant and consistent reduction of the LDL cholesterol. Only very high concentration of atorvastatin and rosuvastatin may produce a benefit in terms of significant reduction of the LDL cholesterol. So this leads to the major problem when using statins, which is the occurrence of side effects, which in more than 30 or 40% of subjects undergoing treatment with statins leads to the interruption of the treatment and to potential consistent occurrence of all the components which may increase the risk, as in the case of the occurrence of diabetes. So, in general, a series of further interventions are required in order to potentiate or to reduce the impact of drugs in treating the high cholesterol. You can see here that the American Heart Association identified the 7 modifiable factors which may affect cardiometabolic risk. And these are represented by diabetes management, control of LDL and HDL cholesterol, blood pressure, cigarette smoking, physical activity, and reduction of obesity. However, the supplementation and the combination of healthy foods with food supplements is today considered the most relevant component of modifiable factors which may affect cardiometabolic risk. But the point is which series of food supplements have to be included into the normal lifestyle in order to potentiate the response of drugs or to reduce the impact in terms of side effect of conventional drugs. And the point is that the major limitation for the use of food supplements in lowering LDL cholesterol is that many of them, which have been studied in the last few years, demonstrated that the response is very weak. The reduction is no more than 15%. There is poor scientific evidence supporting their efficacy in lowering LDL cholesterol. The biomolecular effects are poorly defined, so you have no pathophysiological basis to identify which is the optimal approach via food supplements for counteracting the cardiometabolic risk. We have no data on long-term impact of these components of the diet, and the safety profile is sometimes unclear. Not all the food supplements have to be considered for an absolute safety profile as safe and effective. So, with this in mind, in the last 20 years, we studied novel food supplements and mostly deriving from citrus species, and in particular the one which are originating from the bergamot fruit. Bergamot is a fruit which is growing mainly, as you can see here, in a very selected area of the Calabrian region in the south of Italy, and the major use to date was represented by the use in parfums, which the eau de cologne and many of the most relevant parfums today use the essential oil which is obtained from the skin of the fruit. Many of those products are based on bergamot essential oil, but also the bergamot tea, and in particular the Early Grey, was for centuries characterized by the combination with bergamot. And the fruit and the remaining part of the fruit, and the juice in particular, was used for fruit combinations and for traditional activities which were related to the historical function and use of bergamot in the south of Italy. Is a nice history which I will try to summarize in a very few slides. And you can see here that the natural origin of the bergamot was warranted by manual collection and handling of the fruit, in which the whole family sometimes was involved, and in which the compression of the fruit and the filtration and storage of the essential oil was absolutely natural for centuries. Today, this characteristic is conserved by also in industrial side by selection and washing of the fruit, peeling for obtaining the essential oil, and then the fruit is squeezed and filtrated and pasteurized, and at the end you have the powder, which is the one you can find in tablets or capsules. So the product is absolutely without use of solvents, and the product is absolutely safe. And the principal characteristics of the extract from bergamot fruit are mainly related to the very high concentration of antioxidants and especially on flavonoids. You have a concentration of 1 gram a liter, which is clearly the most relevant you can find among the citrus species, and much more than grapefruit, much more than all the other citrus derivatives. But also the qualitative concentration is very peculiar in the bergamot extract, in which you have high concentration of neoreochitrin, naringin, neosperidin, which represent more than the 38% of the bergamot polyphenolic fraction. But you can see here that many other polyphenolic components are contributing in the composition of the bergamot polyphenolic fraction, or BPF. This is an HPL tracing, which you can see here, in particular 2 peaks of flavonoids which are glycosylated and which are represented, you can see here, by butyridine and melitidine, which are glycosylated flavonoids, which are unique in the BPF, and these are associated to the powerful antagonistic response you can find in the endogenous biosynthesis of cholesterol through the inhibition in a statin-like way of the hydroxymethylglutaryl-CoA reductase, which is the key enzyme in the endogenous biosynthesis of cholesterol. So we have the combination of different flavonoids and antioxidants, which are able to make this product as special and peculiar in the area of natural antioxidants, and as you can see, in the products which are able to counteract hyperlipemia and cardiometabolic risk. And this has been shown in more than 50 papers which have been published to date in top-ranking journal. This is the first evidence we published more than 15 years ago, in which we demonstrated that there is a very specific target in the activity of bergamot extract, which is the receptor for oxidized LDL, or LOX1, which is responsible of the vascular impairment you can find both in animal models of disease and in humans. And in particular, you can see here that when you evaluate the disease, you can see here is the normal blood vessel. This is blood vessel undergoing injury, in which you can see that there is a proliferation of smooth muscle cells, which tend to reduce the size of the blood vessel and to produce something like the atherothrombosis you have seen at the beginning. And it is the brilliant response when you are using the BPF, so the bergamot polyphenolic fraction. And this effect is also related to the change in the oxidative pattern of the tissue after taking BPF. Here you can see that after balloon injury, the same you can find in subjects undergoing the intervention for revascularization in humans, you can see here that there is an accumulation of the nitrotyrosine, which is the footprint for the occurrence of oxidative stress. Here you can see that the bergamot juice is able to not only serum cholesterol and triglyceride, but also serum glucose in epidemic patients, and is a study which has been published now more than 10 years ago, in which you can see that the target of a significant reduction of 42% of the total cholesterol and LDL cholesterol may be obtained directly through the bergamot juice, and alongside with a reduction of triglycerides and serum glucose. And if you look at the responses which may be obtained with the extract of the juice, so the BPF, you may find the same significant response in terms of both reduction of the total cholesterol, LDL cholesterol, triglycerides, and glucose. And here you can see that there is also a slight, not significant, but consistent reduction of body weight, which allows the possibility to obtain the optimal response in terms of reduction of cardiometabolic risk. And you can see here that also other biomarkers such as the atherogenic index, which is really important in animals fed on hyperlipemic diet and treated with BPF, may be obtained with a significant reduction of the biomarkers for the development of atherosclerosis. So the bergamot extract, and in particular BPF, is able to produce a significant reduction of all the components which are leading to the potential enhancement of atherosclerosis process. And also in terms of combination with statins, you can see here that the BPF enhances the effect of rosuvastatin, which is the golden standard of statins in terms of reduction of LDL cholesterol, and also we can see an improvement of endothelium function as indicated by the modulation of the protein kinase B in blood vessels. This is a biomarker of endothelial function. And as you can see here, the bergamot is able to produce not only a reduction of LDL cholesterol and an increase in HDL cholesterol in subjects undergoing treatment with 10 milligrams a day of rosuvastatin, but also we have the additional response in triglyceride, which is the effect which is absolutely required in order to produce a further enhancement in the vascular responses which need to be obtained when a treatment with statin is started. And this is the effect on protein kinase B in human mononuclear cells in those patients in which you can see that the BPF is able to produce a relevant synergistic response. And this is also back to the LOX1 expression. You can see that this is really attenuated by the concomitant treatment of patients with BPF and rosuvastatin. So we have that the placement in therapy of BPF is much more consistent compared to all the other food supplements which have been generated in the last few years, and the level of response is much more like the one of statins compared to all the other food supplements which you can find in the market. The mechanism of action is at different levels, and we have that the most relevant response is in the reduction of cholesterol absorption. Just a few examples. This is a direct effect on the enzyme which is responsible of cholesterol absorption. And after meal, you can see that there is a very nice dose-response curve in terms of inhibition of the pancreatic cholesterol ester hydrolase, which is the key enzyme which is involved in cholesterol absorption. And this is a very clear dose-response antagonistic response. But also, if we look at all the transport system of the lipoproteins in both the liver and in general in other tissues, you can see that the treatment with BPF is able to reduce, to enhance the excretion of fecal steroids, which is a very nice expression of the inhibitory response in pancreatic cholesterol ester hydrolase, but also the biosynthesis of the cholesterol is modulated, and we discussed it previously on the site of the response of the glycosylated polyphenols such as butyrylidine and melitidine, which lead to response in the endogenous formation of cholesterol, which is quite similar to the one you can find with statins. But also we have other pathways which are modulated by BPF. This is the AMP kinase, which is in the liver, and which is the enzyme in which the modulation of both carbohydrates, lipids, and glucose are modulated, and you can see here that this modulation leads to the formation of different lipoproteins, which are used for the transport of the cholesterol in peripheral tissues, but also in the modulation of the endogenous formation of cholesterol. And all those enzymes, APO1, ApoB, LCAT, and PON1 are modulated by BPF, thus suggesting that there is a significant effect and an improvement in the ability of the liver to work efficiently and to release the due amount of cholesterol and to avoid hypercholesterolemia. This is the effect on AMP kinase, in which you can see that after 120 days, the response in terms of overexpression of AMP kinase in patients with metabolic syndrome is enhanced. And a further effect is in the formation of lipoproteins, which is crucial for the development of atherosclerosis. And you can see here that when you have circulating large LDL particles, you have a protection in terms of development of atherosclerosis. And the small dense lipoproteins are the ones which are characterizing the pattern of very dangerous response in terms of lipoprotein formation, which leads to atherogenesis and oxidative stress. And this effect is clearly correlated with an increase in mortality, in which you can see that the small dense lipoproteins are associated to a reduced survival correlated to cardiovascular disease, and these are all the conditions in which small dense lipoprotein may produce consistent damage. And if we treat subjects with BPF, you can see that you have an increase in the size of LDL. So you have not only reduction of serum cholesterol and triglyceride, you have not only protection of blood vessels by increasing and improving endothelium performance, but you have also reduction in the size of LDL, an increase in the size of the LDL, which leads to protection against atherosclerosis. And finally, the most relevant response is in terms of oxidative stress and inflammation in the liver, in blood vessels, and this occurs through a potentiation of the endogenous antioxidants such as manganese SOD, catalase, and GSH peroxidase. And you can see that this effect is crucial since a reduction of the endogenous antioxidants enhances the oxidative stress, which is related to an increase of peroxynitrate formation, as we have seen in the previous slides. And if we look at the level and the potency of BPF compared to traditional antioxidants, such as vitamin E, which is the most famous one, and other chemicals such as metalloporphyrins, which we use in the last few years, and which are chemicals, you can see that BPF produces an excellent performance in terms of antioxidant activity, which occurs at very lower concentration compared, for instance, to vitamin E. So it's a complex and multi-action response, which at the end leads to a reduction in lipid peroxidation of the blood vessels. Going to the conclusion, our studies demonstrated, alongside with all the other studies you can find in the literature, that lifestyle changes and natural antioxidant supplementation is able to counteract cardiometabolic risk, to improve endothelial function in patients with hyperlipemia and metabolic syndrome. Bergamot polyphenols and BPF in particular have been assessed to represent the most powerful natural source able to antagonize oxidative stress and inflammation in blood vessels of patients with moderate to high cardiometabolic risk. I'd like to stress this concept since not too many of the products you can find in the market have been demonstrated to produce an impact in high cardiometabolic. And finally, the combined effect of BPF on serum cholesterol, triglycerides, and the properties to counteract insulin resistance makes this natural extract the most effective and safer resource for the prevention of vascular damage accompanying metabolic syndrome. I am really sorry for the difficulties, but I hope that everything has been represented in the due way, and now I think that we can answer to the question in order to clarify eventually which are the most relevant points of this presentation. Thank you very much for your attention.
Thank you so much, Professor, for hanging with us and for such an informative presentation. Did we want to go into the Q&A at this point?
Okay.
Excellent. Okay, so before we get started, I would like to remind our audience how to submit questions. You can submit questions by typing them in the Q&A box, which can be found on the right-hand side of the presentation window. And again, any questions we do not get to during this presentation, we will address at a later date. But on to our first audience question. What is the most consistent difference between BPF and other natural antioxidants?
Okay, thank you. Well, one of the most relevant aspects is represented by the very high concentration of polyphenols compared to all the other species we have been able to study in the last few years. It is not only, as you have seen, a matter of quantity of polyphenols, since you have also specialized polyphenols as the glycosylated one, which produce a statin-like response. The most relevant aspect is that this occurs without any of the side effects you can find when using statins. And this suggests that this is a very gentle way to inhibit the endogenous biosynthesis of serum cholesterol. So the other aspect is that the activity occurs at multi-level at different stages in the development of atherosclerosis. And this activity is like the one which is requested by the modern approach, synergistic approach to the treatment of high cholesterol and hyperlipidemia.
Excellent.
Okay.
Thank you so much, Professor. On to our next question. What are the mechanisms that lead to vascular protection found in individuals who supplement with BPF?
Well, the first basic mechanism is mainly represented by the restore in endothelial response, which is the most relevant and consistent aspect you can find in any of the patients which have been treated. This extract has been studied in more than 20 clinical studies all around the world, and many of them in top-ranking. The results have been published in top-ranking journals, and more than 2,000 patients in clinical studies. So this means that there is a consistent experience in managing this product, and the major characteristic is the antagonism in vascular inflammation and in endothelial dysfunction.
Excellent. Thank you so much, Professor. But unfortunately, we are out of time for today. At this point, I want to thank the audience for hanging on with us and for attending and participating in today's event. And I'd also like to thank our sponsor, HP Ingredients Corporation, for making today's educational symposium possible. We would like to ask everyone in the audience to participate in a brief survey from our sponsor, which you can find on the right-hand side of your screen. You will be receiving an email alerting you when this webcast will be available for replay, and we invite you to forward that announcement to your colleagues who may have missed today's live event. Again, thank you so much to all for joining, and we'll see you next time. Goodbye.